Hey, this is Dr. Ben Morrill. Welcome to episode 14 of Reptile Genetics Weekly, and we are live this time. And for those of you that are live on Instagram, you'll be able to see me. If you want to see what else is going on, we're also on Facebook and YouTube. So as always, we'll start with a results update. So we are currently doing the biggest run we've ever done. So we have a very sincere thank you to give to all of you. It's been a very busy last week or so. It's been a lot of fun though. It's cool to see so many tests, so many people utilizing this very thing that Shawn and I thought would be helpful. It's really nice to see that it is. We've got lots of samples coming through. This next weekend is when we will start sending out results. So probably I'll be able to start analyzing on Friday. So probably Saturday, Sunday, Monday would be my guess. We'll have the next results going out. So yeah, Kayla, and we've got Shawn here too. How are y'all doing?
Hey, we're doing great.
Hello, everybody.
Hi, guys.
Hippo Herp says, caught you live. Hello.
Hi, Hippo Herbs. Welcome.
Looks like we've got several on Instagram as well.
Good to hear. Yeah, leave us comments and nice alive. Hi, Morph Wizards. I don't think we've seen you here before either, so welcome. Yeah. Just getting started, so you're here in time. We've got 16 people in the stream right now. Awesome. So we got Reptile Creation checking in and Matthew Kriego, IMC Reptiles. We got everybody. There, and we're getting questions. Awesome. So we got a few updates. Do we want to get with those real quick? Or
let's go ahead. So now people are jumping in. Let y'all know that we wanted to announce a couple of big shows that we're going to be going to.
Yeah.
And so I'm going to go next month to the Pomona show in California, to the Reptile Super Show. I know we've got several people that just vended the Super Show that was in Anaheim. And I saw lots of good pictures, lots of people there that I've talked to online and haven't met in person yet. So I used to go to the NARBC Anaheim show every year when I lived on the West Coast. I went with Justin Julander, who's Australian Addiction Reptiles. The two of us would go to that Anaheim show every year, but I haven't been in many, many years. So I'm excited to be at the Pomona show. So if you're going to go, say what up, and look forward to meeting many of you and talking in person.
And we're hoping to get some merch to bring there too, so you can actually see it in person, buy it there. So go see our boy.
Yes. What you want to see, what merch you want.
Exactly. And Shawn's actually going to one as well. So Shawn, do you want to talk about that show?
Yeah, I'll be at the Daytona show. This is the first time I've probably been there in 10 years or so. I'm originally from South Florida. I used to attend the Daytona show quite a bit. But I live in Texas now, so that's a rough one for me to make. So it's cool to be able to have a reason to go besides just wanting to be around other reptile nerds like myself. I get to actually go talk about what we're doing and how much fun we're having over here. So
yeah, there you go. Sweet. So we got those shows. Now this one I'm going to hand to Shawn as well, because we are sponsoring a semi-pro MMA fighter.
Yeah. So look, this is not as big of a deal as it sounds, but a lot of people may know, or may not know rather. Ben and I both studied jiu-jitsu. We have for a while, so we're both fans. We like to get on the mat and try and choke people out occasionally. So we like to watch fights. We like to support fighters. Caddy Strite is a relatively new professional fighter. She's got a record of I think 1-2 right now. So she's got a fight coming up this weekend, and I've personally sponsored her since she started, and this will be her second fight that RGI has officially sponsored her. For all of our friends out there that are MMA fans and maybe jiu-jitsu practitioners or Muay Thai or something like that, give her a shout out and a thumbs up and tell her you're rooting for her. She's got a big fight coming up this weekend.
Yeah, Caddy Strite. Awesome.
Cool.
Sweet. And this is just a reminder again that we do have our merch store up. All of the proceeds for these are going right back into further morph testing. We do have, so we've got women's racerback stickers and hoodies, regular shirts, ball caps as well. And also we have the logo ones, and then these like the funny logo, DNA test results, reptile nerds. Down at the bottom there it says, we didn't need a DNA test to know that we're reptile nerds at heart, but it's best to show it off. We like cute stuff like that. If you think of any yourselves, please send them our way. Maybe that'll become like a little giveaway or swag bag type of thing at some point. So, we like cheeky things and we like cheeky ideas.
Shawn's the one that can make y'all a promise. We'll put him on the hot seat, see if he wants to say what prize we'll give someone if they come up with
yeah.
That's not 100% fair. Look, here's the embarrassing truth, right? Like I actually, my college, courses, my degree was in creative writing. And I like to think that I'm a pretty good writer, and I can't come up with anything creative and funny for our business. Kayla came up with this one, and it's great, but I can't figure out any more. And that's really unusual for me. So I really personally am just sort of reaching out to everybody. And those of you that are watching, forward this to your friends or just mention it to them. You can have them message us through our Facebook page. I see that quicker than most other people. They can just send me a little one-line slogan if they think it's a good idea, or you can send me a piece of artwork that you think is appropriate, with our logo in there somewhere. Even if it's not really well done artwork, I'll pay to have it really well done if you just have a really good idea. I want to get some really cool shirts made, stuff that's fun to wear and doesn't just show off the brand, but is also hilarious and funny and, pokes fun at all of us as a unique hobby, right? So get creative, guys, and send me what you've got. If it's a neat logo that I can use somewhere, maybe we'll throw you a free test or, a t-shirt or, a bunch of stickers. If you send me a really good piece of artwork that's really well done, let's just say that the offering will be a lot more substantial if I don't have to pay somebody else to doctor up what you've already done for us. Then, so yeah, please, you guys get as creative as you can, because I'm struggling over here, and that's unusual for me, but I have not been able to come up with something.
Yeah. And also that would definitely, like, if that were to happen, we'd definitely give a shout out to whatever artist
absolutely
helped out.
Yeah, absolutely.
In addition to the commission.
The sooner the better. These are shirts that we don't really want to make available through the merch site. We want to make them available only at shows, so that we know that you came up, shook our hand. If you're wearing our shirt, it's because you either bought it from us or we gave it to you, just because we appreciate you guys doing business with us. So any of that kind of stuff that's unusual from our normal merch stuff is what we want to be giving away at shows. So send us your ideas and send them quick, because I need to start making shirts here in about a week.
Yeah. Or tell us if you want just like a really like a really high quality picture of just Ben's face cut out with the hat. So, just let us know if that's something you're into, if you're into that sort of thing.
You have one comment on Instagram. Nevermore Ball says, just took a DNA test. Turns out I'm 100% reptilian.
All right. That's a start. Exactly that kind of stuff.
Very cute. I like it. All right. So Q&A time?
Yeah. And we're getting a lot of good questions. I see you're highlighting some. That's great. Thank you so much. Keep sending comments in. I see High Desert Python says he got his hat. I'm glad you got it. I know that the projected delivery date was like a month away or something. I'm glad it was faster than that.
Yeah, no, actually, I've noticed on my end on the merch store, like they tend to print about every 2 days. So it doesn't seem so unless they have a crazy high volume for some reason, the shipping times don't seem to be too bad. But yeah, let's get to some Q&A stuff. We've got from Scales and Tails Breeding. I know you were mainly focused on ball pythons, but I do have some bearded dragon sheds I can send you to develop later tests. Would you want them?
Yes, for sure. So bearded dragons are a little interesting in their sex determination. So I'm not interested in designing a sex determination test. For any of you that are interested in the super nerdy stuff, let me know. We can go into that someday. But they have a very interesting system where it's based on genetics, but it can be affected by extreme temperatures where they become just one sex. I think if it's too hot, but I can't remember for sure. But anyway, I'm not interested in designing a genetic test because it wouldn't always be correct. It would depend on incubation temperature. But yes, for morphs for bearded dragons, yeah, send them in. We're very interested in that.
Good to know. We have from the Hitomi boy. Do you have an update on when the Axanthic line testing will be available? I assume for ball pythons and
Yes.
all the Axanthics.
Yeah, so for Axanthic in general, for ball pythons, that'll be first. And I would imagine it will be sometime in the next month, 2 at the very most. But yeah, it's kind of in that stage where if what I have is going to work, then it'll be real quick. If what I have doesn't work, then I might have to go back a step or two. So, but yeah, we're very close for VPI Axanthic and TSK Axanthic. And I've had a few people message me just this last week about getting some MJ Axanthic sheds to me. It might be enough to where we can start sequencing, but that would be, since we haven't sequenced, that would be like at least 4 months out. But the other cool thing is we have a whole bunch of Axanthic sheds from Drymarchon. So the same genes that indigo snakes are in. I think it's actually the Crebos that the Axanthics came from. But anyway, we also have carpet pythons, and I think it's Bredeli. Can't remember for sure. But anyway, 2 different carpet python kind of complex animals that we're getting Axanthic sheds from. And then also from Split Rock Reptiles in Texas. I was just talking with him again this last week. He's sending Axanthic black-headed pythons and maybe some hypos and some normals. So it would be kind of cool if a year from now, if we can kind of talk about Axanthic in reptiles, especially in snakes, and what's different and what's similar. But we're building kind of a cool story that we'll be able to tell. But for ball python specifically, for VPI and TSK, the most common ones, those are coming pretty quick. And then I hope in the next month.
Yeah, that'll be really cool. We have from Morph Wizards. I don't think I've heard you discuss this before, but have you worked at all with leopard, pinstripe, or other morphs that people debate if there's a super form?
Yes. Yeah, we're actually pretty close with both leopard and pin. I would imagine sometime in the next couple months, I'll get at least one of the 2. But that is definitely dependent on how crazy things get in baby season. I might have to stop working on new tests for a month or so just to be able to handle the volume. So that could make it take a little longer. But for both pin and leopard, we are fairly close. And we're definitely working on others, like that maybe as a single gene test aren't super interesting to people, but as a panel for, combos or for people that, pick up an animal and don't know at all any background, they just want to have a panel test done. We're going to keep adding more and more.
But I do want to say, and I don't mind, folks in the hobby knowing this, that one of the things that holds us up, we've actually got a lot of sheds out for sequencing, sheds that will probably close out a lot of these morphs that we're pretty sure we're close on, but we just don't have enough data to be sure. And we have to send those out to a much bigger lab, and we're sort of at their mercy. And that sucks, but unless anybody wants to throw $500,000 at us to buy our own machine or more, it's probably more expensive than that at this point. That's one of the holdups is we put these plates together. We've got all this data that we're hoping to get back. Sometimes we get it back quickly. Sometimes it takes 3 months to get back. We are a very small business compared to the pharmaceuticals and, the I don't want to say I almost said urban, plant industries, right? And all of the flower people and all, they all do a lot of genetic testing too. And we're getting runs on the same machine. So that's part of what the holdup is it's not our inability to get the work done. So sorry, we can't get the data to us fast enough. And we're currently looking for another lab to help us with that. But we're small potatoes in a big world that's discovered genetic testing is pretty big deal. So we're lucky to have the partner that we do. However, if we catch them in a good month, we get our results really fast. If we catch them when they're busy, we get told to wait, and that's just how it is. That's what correct me if I'm wrong, Ben, but that's what Axanthic is waiting on, right? Is that final confirmation, that last bit of data from this last run that we did. We submitted those samples 6 or 7 weeks ago.
Oh, more than that.
Yeah, they've been in for a long time now. So
probably 3 months, 2.5 months, something like that.
Right. And so that's what we're dealing with is we prep all of these things. We think we've got everything figured out, and we send it all off for the higher quality sequencing run than we're capable of doing, and then we just wait. And unfortunately, that's what we're doing right now, is we're waiting. That's why we keep, it'll be another month, because unfortunately we'll get told on the first of the month, well, guess what, guys, it's going to be another 2 or 3 weeks. So, and we've been told that 4 times now. So it is what it is. We're doing our best, but when we were big enough, hopefully we'll have our own machine to do all of this stuff, but we're just not there yet. So,
and even as we get bigger, we become a more important customer.
That's true too.
Happen a little faster. So we were a nobody 2 years ago, and now they at least know our names, so we get some stuff done. But
we send in 4 place once every couple of months. You know, other people send in 400 a week, so we're just not big players as far as that. Anyway, go ahead.
We're coming.
Yeah, it's all good.
We'll get there.
Okay, this is from Reggie's Urban Jungle. Are you close for a monsoon test?
So I'm getting to where I have enough sheds finally. So those will go to sequencing fairly soon. So that's another one, probably like a 4-month, because we need to do the sequencing, get it back. Then I do my analysis and design, and then validation of those tests. So monsoon's still a little ways out, but I think we do finally have enough sheds. But if you have a, homozygous, a visual monsoon, and you want to send a shed in, right now is the perfect time to do it, because every one of those I get will help increase my chances of finding it faster.
Yeah.
So even if it's a het, that's great, but especially visual monsoons, right now would be perfect. If I get that shed in the next week or two, then I can make sure it makes it on that big sequencing run.
Yeah, actually, that's better than the last stream when someone asked about monsoon. We didn't have enough sheds for it yet, so now we do. That's thanks to you guys sending those in. So that's great.
Yes, I don't know the name of the person, but somebody sent a whole bunch to Canova, and they forwarded them on to me. So I have like a dozen from somebody. So whoever it is, if you're listening to this, you have to chime in, or I'll have to ask Justin who it was. But that was awesome. That's very helpful.
Yeah, and if you don't know how to get there, here's the clip that's covering Shawn's face. That shows you how to get to our page, where to send a shed in. It's on our website, and you just scroll right down on there. It tells you what to do. Now we have another one from Jorge Gonzalez. That is, what about new tests for more recessives that are becoming more popular, like cryptic, paint, black axanthic, possibly too?
So cryptic, we're fairly close on. I could still use more sheds. I might have enough in these results that we're trying to get back. I'm kind of on the edge for cryptic. I had something I thought was going to work, and it didn't work, so I had to go back. But this next batch of data, I'm going to try again on cryptic. Paint, I have very few paint samples. So if you have paint, that definitely would be a great one to get in. Same with black axanthic. I think I have 5 or less of that. So get those into me. That's really what, Kayla was actually asking earlier, how do we kind of decide what we're doing? It really boils down to, we can only do what people send us sheds in for. So I can't design something, what it's different from a normal, unless I have DNA to look at to be able to see how it's different from a normal. And so, yeah, those sheds, that's the biggest thing. We do have a comment. Let me throw one off from Instagram. This is Hades Herb. Hades Herb says, I would be interested in being a Patreon, especially if it helps with getting gene tests, maybe as a member pricing or something like that. We've discussed that. We haven't made it happen, but definitely if there are people that would be interested in that will for sure help. The research dollars are pretty big. That's where everything goes right now. So like we say with the merch store, anything from that is just going to go right into testing. So if that's something people are interested in, I did spend a little time on the Patreon page. I didn't spend enough to get something started. But especially if someone has an idea what would kind of be a good thing for us to offer, I think it's pretty common to have 3 different tiers and something that you're offering. Mm-hmm. If someone thought through and kind of had some ideas, it's just a time thing to be able to get it set up and have something that's going to be beneficial for you all and be able to get us more research dollars. That would be awesome.
Yeah, definitely. Let's see. We have Oh, from Clover's Reptiles. This was something we answered at the beginning of the video, but I think you might have come in right after that. Shawn will be at Daytona, so go and catch him there. We have from Lori Torini. Hi, Lori. Yes, speaking of non-Python Regius, any news about Morelia tests or other Python species?
So right now, the only one that that I have some keepers talking to me specifically about is axanthic in carpet pythons. It wasn't Bredli. It's poplin carpets and coastal carpets. So apparently, axanthic in both of those species is compatible. And so I'm going to get samples of both what we used to call Iriangias, now they call them poplin carpets, and then the coastals. And so we'll hopefully have enough over the next. I think some people are waiting for clutches to hatch, and they'll have a bunch. Since carpet pythons have massive clutches sometimes, especially a big coastal female, you can get 30 or 40 babies sometimes. So we'll all of a sudden have a whole bunch of sheds, hopefully over the next month or two. And I have started getting, I had a really good week this last week getting blood python sheds in. Got a whole bunch, 2 or 3 people. I probably got 30 or 40 blood python sheds.
Yeah.
I got a few retics. That's probably the species people have asked for the most, but I've gotten the least sheds is for retics. Mm-hmm. So I could definitely use more retic sheds. And then berms. I'm still getting some more berm sheds in.
Two things. One, along with carpet python, several other species like bloods, we're pretty confident we're going to get sex determination across multiple pythons when we get this next batch from the big lab we were referencing earlier. Secondarily, I got an invite from Bob Clark to come up and collect all the sheds.
Oh, excellent.
Yeah, I was wondering because I think he's not too far from you, is he? Right, Shawn?
He's not. He's just a few hours north of me in Oklahoma. So I'm probably going to road trip up there in the next few weeks and spend a day taking sheds and scale clippings and whatever else I can, swabs, whatever else I can do to get as many samples from as many individual animals as possible. Like I said in our last live cast, a month ago, we will travel. If you have a big enough collection that's representative of lots of mutations, we're happy to drive or even fly out to come visit you if it'll help us gather the data that we need.
Yeah,
definitely.
Shawn, you should get some awesome footage while you're there, because that's a lot of cool animals you'll be seeing.
I have to ask permission, but we'll see.
That's fair. I'm kind of jealous. Anyway, next we have from Hippo Herps. Will there be Western hognose morph tests developed?
Definitely will be. And I have had a good amount, not a ton, but I have had a decent amount of hognose samples come in. Corn snakes, I've had quite a few. There's some people that have sent me 30 or 40 at a time. So corn snake samples are coming in really well. So yeah, I mean, all this stuff takes time. Those of you that have kind of been following this, all the ball python work really started for morphs started a year and a half ago or something.
Yeah.
So, I mean, it takes time. So that's why we wanted to get the word out. We want sheds from other species so that we can get the ball rolling. It's not going to happen tomorrow. I know I have all the time people, they're like, hey, I sent in my Western hognose sheds, are you going to have that test ready? And it's going to take months, if not a year or two, but the ball's rolling. And as long as people find this testing helpful and are paying for tests that we have developed, we'll turn around and use that money to develop more. But that's definitely what we're excited to do.
Yeah, I feel you, Hippo Herps. I'm a hognose keeper too, so I feel you. We have from Sam Fisher. Do you plan on doing Orange Dream or Super Orange Dream tests anytime soon?
Yes, very close on that. It would not surprise me if I get it in the next month, but if I do need more data, if that doesn't work, then it could be 2 or 3 more. But yeah, it looks pretty good that I'll be able to get that.
Yeah. All right, and we have from RSB. This is more of a comment than a question, but there needs to be a standard lexicon for the polymorphic genes, as in DGAABB could be DG alpha, and then DG AABA, just look at the stream, sorry, Instagram, could be DG Bravo, and so on. Do you have any thoughts on that from your insight, Ben, or is that something the hobby's just going to have to deal with?
I think of it from, how we learn in genetics textbooks, and I don't know how helpful that is to other people. So I'm probably not the best person to ask about that.
Okay.
I do believe that we'll see over time. Like I've seen some people start to say, hey, tested het for DGA, tested het for DGB, or tested het for both, So that's one way to say it. But yeah, I think that the amazing amount of sheds we have coming that have pictures attached, and some bigger breeders as well, or sending a whole bunch and have shed or have baby pictures, and some of them will have pictures when they're a little older as well. I think that's going to be huge to be able to figure out maybe a DGC or a DGD. That'll help a lot in telling that story. What were you going to say, Shawn?
I was going to say thank you to the hobby. So we're just a few weeks out from when we did the free Desert Ghost testing, and we made it clear why we were doing that, is we wanted lots of sheds to start coming in. And I have in hand probably 120 samples with photographs already in 3 weeks.
Excellent.
Yeah.
So it's been 3 weeks, 2.5 weeks. So it is definitely working. And, if everybody that sent me an email to register sends me 2 sheds, we're going to have 1,000, maybe over 1,000 sheds to test. So
wow.
We have a lot of work to do. And so far, the response has been fantastic. Everybody's following the instructions, sending me the data that I need, sending me really good quality photographs of animals that are either related to each other or sire to offspring, that sort of thing. So we're really looking forward to what the future holds there.
Yeah. Oh, we have a visit from my cat, Potato. Look at her back there. She's beautiful. She's also a jerk.
He's a cat.
Yeah, exactly. Let's see. RSB says, I for one appreciate everything your team is doing. I'm happy to be on the ground floor. We appreciate you, RSB.
Yes, thank you.
And for all the sweet things that you guys are saying. That one just jumped out at me and I was like, oh, that's nice. We have from I think this was in relation to the MMA sponsorship.
Oh,
I need more grappling matches for
like hobbyists are gonna roll? Really? Can we do that?
Wait, what is this? I'm grappling matches.
Is that what he's talking about, Ben? Do you know what he's talking about?
I haven't heard of it before, but that'd be fun.
No,
that would be fun. I'll roll for charity. Heck yeah,
I'll do it.
It sounds fun.
I've gotten choked lots of times. If I have to choke somebody, that's even better. But if I need to get choked for charity, I'll do it.
No gi though. No gi for me.
We have from Clover's Reptiles. Generally, how many sheds do you need per genetic per trait to make a new test?
It definitely varies. It'll kind of depend on. I guess the simplest way to say it is we want to capture as much genetic variation other than the actual mutation we're looking for as possible. But then also be able to have animals that have it and do not. So if I had 20 sheds and they all come from the same male and like 2 females, that's not as helpful than if I have 20 sheds that are homozygous and they're from 20 different pairings. So not every shed is the same value, but on average, I would say 20 is a good starting point, especially if they're homozygous for that mutation, whether it's an incomplete dominant or a recessive trait. If we have 2 copies, we know that the only signal we'll get is the mutated signal. That helps us design faster. But even hets are helpful. But yeah, 20 is a starting point. Sometimes I get it. 40 to 50 or 60 is what we really like to have. I think for clown we had like 60, and that was really nice. And the fact that it was only 1 mutation and that it was simple recessive, everything that we expected and as simple as it could be, it made clown real fast.
It was in a real sweet spot from the sound of it.
Yes, I wish they were all like that.
Yes. We have from Troy Schroeder. Are you working on a Mandarin test?
I have very few Mandarin sheds, so I would definitely love to get more. I have a handful. But yeah, I would love to do that. I think that would be helpful for people. From what I've heard, there's questions sometimes. And I know some people have done some testing to find out whether their mandarins have het hypo or hypo in them. And so hopefully that's kind of helping to clear up some of the questions that go along with mandarin. But yeah, I would love to get more sheds and be able to develop that. That'd be great. They're beautiful, that's for sure.
Oh, I just remembered something related to, like, back up like 2 questions. This was a Shawn question. So earlier on Facebook, we had Larry Fish comment, when will we start getting replies regarding when to send our Desert Ghost sheds in if we emailed you when asked? So this was regarding the event where we just had like a Desert Ghost testing blitz, and you're supposed to send a thing to Shawn. So
you had 3 days to register. About a week after that, maybe 4 or 5 business days after that, I sent out a pretty large constant contact email list to the hundreds of people that replied within that 3-day period. A few of you, if you did not get that email, you need to message me through the Facebook page and let me know. Larry's one of those guys who, for whatever reason, his reply or his email got sent straight to my spam folder for no reason at all.
Larry.
That's where I found it. So he did not get the reply email with all the instructions. So if you're one of those people that didn't get a reply email, as long as I can confirm that you sent me an email in that 3-day waiting period somewhere in my spam folder, or maybe accidentally deleted it, you're good to go. I'll send you everything you need. Just let me contact me through Facebook.
Awesome. Thank you, Shawn. We have from Forward Motion Reptiles. What about a test to set the debate to rest on hurricane and trick being the same or different?
So I know that I have a few hurricane sheds. I don't know if I have many trick at all. But yeah, I would say both of those, I need at least a few more for hurricane and several more for trick. But yeah, that's another one I'd love to nail down for sure. I just need the sheds. If any of you know somebody that's got several hurricanes and tricks, I would love to get those sheds.
Yeah, go get them. We have from Scales and Tails Breeding. I always send full sheds when testing, just in case. Do you use that extra shed to help develop tests?
Yes, as long as you have notated on there all the morphs that are present and all the morphs that are possible, then we do a pretty good job of figuring out a way to file ones that we feel will be used fairly not too far in the distant future. So if we're going to use it fairly soon, I do a pretty good job of filing it so I can grab those anytime. But I also go back. I have boxes and boxes of sheds. I go back and dive in. If there's something I want to get, a dozen more of, I'll go back to sheds that I got 3 or 4 or 5 years ago, and just sent a whole bunch of various python species this last time to get sequenced, and some of those I got 5 or 6 years ago. So yeah, I don't throw away sheds.
Very good.
But as far as the full shed, you really don't ever need to send the full shed. So the size of a quarter to a silver dollar, that's like 30 to 50 tests that I can do on that. So really, a silver dollar is definitely as big as you need to send. It's more important that it be completely dried out before it goes into the plastic bag, and that it be clean. So just pick the silver dollar size piece that is the cleanest and get that completely dry, put it in a Ziploc, and I promise you, no matter how many times I need to come back to it, I'm not going to run out.
Perfect. From Lori Torini, we have, what about sex determination on Morelia and other pythons? I know you mentioned that before. I don't know which ones.
So green tree, we have the most data coming back. As soon as we hear back from the lab, like Shawn was talking about, people have the real big, expensive sequencing machine. As soon as we hear back from them, that's one of the number one things on that run will be a whole bunch of green tree python known males and females. And so we hopefully can get green tree python. That's, like we talked about before, that's the one that Shawn and I wanted. That's the whole point why we started doing this, is because we wanted a green tree python sex determination test.
That was the original test idea. That was the first one that we talked about 11 and a half years ago. That was the test.
Like the original, that's like our original, like, holy grail white whale thing that's been eluding us.
Yes.
The conversation started with, how come we can do a sex determination on a parrot but not on a snake? And just no one had done the work and the research. And I'm like, well, man, back then, and even some to this day, we're big carpet python guys. Ben and I were back then. That's how we met. And yeah, it was the green tree python test. I'm like, man, nobody can sex these things. Everybody's having to grow them up and then try to breed them first. And that just seems silly to me when you can take your parrot down to the vet for $50, find out if it's a boy or a girl in a week. So that's how this all started. Believe it or not, was that conversation right there.
Yeah.
Yeah.
So yeah, we would love to have it, and I'm pretty confident once we nail down green tree pythons, the carpets will be pretty easy. But maybe I'll be surprised again. I thought when we had so much information about the ball python that green trees would be pretty easy. From that, I'd be able to adapt it and make it work, but I haven't been able to. So we'll see. But carpets and green trees are a lot closer together, and I do have some carpet python sheds on that run too. So I'll have a pretty good amount of information to work with. So hopefully that's coming soon. And I will be testing out on bloods and short tails and retics and berms as soon as I can. We have so many sheds coming in for people. It'll take a little bit of time, but yeah, we are on it.
That's ridiculous. Sorry. I had to highlight that.
Nice, Antoine.
We have from Jorge again. Would homozygous clown / cryptic, aka Krypton, sheds be okay?
Yeah, that's definitely fine. As far as designing the cryptic test, yeah, that's still helpful.
Yeah, I guess since we already know what to look for in clown, then it'd be to the exclusion of that to find cryptic, I guess.
Yes.
From Warfell's Morphs, I hope I'm pronouncing that right. Would you be able to test sheds from jungles, carpet pythons to see or any other localities to see if they're pure or mixed with other localities?
So that's something we definitely want to work toward. That takes a little bit of time. So we have to have a genetic pool that's known. So we'd have to have like a pure jungle genetic pool of, I don't know, 3 or a dozen. It'd be nice to have a dozen that we know are pure jungles, and be able to compare that to pure coastals and blah, blah, blah. So it would take a little while to get to that point, since it's difficult to get samples from Australia. We have to talk to zoos that have older animals or have a way to be able to get samples in or something like that. But yes, we definitely, that type of test is done all the time. And actually the forensic test that they do at a crime scene is the same kind of a thing. They can tell you what kind of ethnicity and all that kind of stuff. They can tell that from those types of tests. They can tell you it's a male, white male, blah, blah, blah. They'll know that just from that genetic test. And that's the same kind of thing we'll be able to develop. But we have to have that genetic pool of what makes a jungle and what makes a coastal, what makes a diamond. We don't have that right now. But yeah, once we have that, then that would be a pretty simple test.
Yeah. And I know we've talked about in the past, like probably our first or second episode of Reptile Genetics Weekly, we talked about localities for like chameleon species and how keepers of those animals would be interested in it. And I know there are a lot of like locality people in the rat snake hobbies, like I'm repping Dark Horse Herpeticulture today. So, we've got stuff for our locality folks too.
Oh yeah. Scrubs.
Or hoping to.
It would be fun to do a project on like just Lamprepeltis, Lamprepeltis hobby, and just see how mixed, Honduran. Yeah. I breed Honduran, so I hear people call them hobby Durans.
Hobby Durans, yeah.
For sure, a lot of them are likely mixed, but it would be a really fun project to be able to go through and go back to some of the older animals, and maybe some samples. People have animals that passed away 10, 20 years ago in their freezers still, and stuff like that. Be able to see kind of how the captive population, the genetics have changed in different species groups. That would be another really fun project.
Yeah, it would be. We have from William Philippek. Does it look like any other incomplete dominant / recessive traits might end up being polygenetic traits?
Is certainly possible. I don't see any evidence yet, but I wouldn't have guessed DG was either, so that doesn't really mean anything. It certainly can happen. But yeah, I haven't, every time we've done the plots where it shows across all of the chromosomes, we always get one peak. Desert Ghost's the only one that gave us 2 big peaks. That's the only time that's ever happened. So,
Okay.
so I haven't seen it yet. Haven't seen evidence of it yet, but it certainly won't surprise me if we end up seeing it at some point.
Troy Schroeder would like to know if you guys are joining him for breakfast again this fall.
Sounds good to me.
Yeah, Ben and I both have a commitment to be there at the fall show. That's like our anniversary show where this all came to life, right?
Which show is that?
The October Tinley. Is it
Oh,
October? It's October, right?
Tinley. Cool.
Yeah. And so we went out with Troy and several other guys to a pretty sweet little breakfast place that they found, and I guess they've been going to year over year. And we went a year ago in October, and then I went again to the spring show, but I wasn't able to stay Sunday morning. I actually drove from Texas, so I hung out all day Saturday and then got up early Sunday morning to come home. But every October for sure, Ben and I will both be there. That's like, I don't know, it's our anniversary. That's our sweet anniversary, the two of us.
Yep, that's when we started working on this.
That's right.
It's a date.
Yeah, I guess it's getting close enough to October. We should bring that up. Yeah, we will for sure both be at Tinley.
Yep.
Yeah. All right. Now we have from Snake Loop Reptiles. Anything regarding the blue-eyed leucistic complex? Blue-eyed or black-eyed?
Yeah. So the first blue-eyed and black-eyed tests we'll likely have will be Mojave and Fire. I'm looking at others as well, but those are the 2 that I have the most sheds from. So those will most likely be the first that we'll get. And I don't know if Dr. Hannah Siddell listens to this, but if you do, we talked briefly before, that might be a good project if she's interested, to be able to take some of the location information that we have from what we've done, because the angle that we take is different than the angle that she takes, and it would be really cool to be able to work on either the black-eyed or the blue-eyed with her and figure out everything we can, kind of like what she did with Yellow Belly. But we'll see. Any of you that have contact with her, you should put that bug in her ear, say, hey, you should work with RGI, and let's get one of these blue-eyed or black-eyed Lucy complex figured out. That'd be a cool project too.
Yeah. We have from Forward Motion Reptiles. Okay. Sorry. I was wondering, at first when I read this, I thought 2-head was a morph, and I was like, I've never heard of that in ball pythons, but okay. Headed to you as soon as this girl has her prelay shed. But I think he means 2 sheds.
Excellent.
That's what happens when I'm listening to 2 things at once. Well, listening to one, reading in another. Yeah. Anyway, you're awesome, Forward Motion Reptiles. Keep it up.
Yeah. Any samples from 2-headed snakes would be cool too.
Actually, yeah, that would be fun.
I don't know if we'll learn anything, but that would be nice to have that in the database.
Yeah. Also from Antoine at High Desert Pythons. Last year I did a match with John Feely. I think this is about the grappling events. Got over 10K for US ARC. Awesome. This year we're having multiple matches at Tinley, so he's going to send you a DM, Ben. Awesome.
Yeah, that'd be fun. I haven't done a competition ever, but I've been rolling for, I guess, 3 years, something like that, in a class setting. But yeah, I would certainly consider it. I would too.
Shoot, I'd like to see that. That sounds fun. We have from Zeus's Serpents. Are you working at all with Monarch?
Monarch is very close. That one we should have in the next month. Like, that's another one. As soon as we hear back from the big lab with the big machine, as soon as that data comes back, we should have Monarch.
Sweet. And with Morph Magic Exotics, will there ever be a database to know if snakes we purchase have tested, or like if snakes we have purchased have tested before, I assume with other breeders?
Yeah, so that's something we're working toward, and maybe I'll let Shawn kind of talk about as much as you want to about that, because you know that's one of the subjects I go off on, so it's safer for you to talk about it.
Honestly, I sort of missed the question because I have to reread it. Hold on. Will there ever be a database to know if snakes we purchased have tested our snake before? Wow.
So that's the other thing that we talked about from the beginning. We want to be able to have a database where information's recorded, and there'd be, ancestry information, that kind of stuff.
Well, and one of the things that we talked about in our last live stream was that we're going to be doing the DNA registry in place of our panel in the future. where, you'll get panel results, but we're also going to have a electronic version of the fingerprint for that animal. And a lot of breeders have gotten back to us with specifically this sort of an application where they're saving baby sheds to every baby that they produce, saving that first shed, labeling it, and just filing it away, so that in the future, if they have a problem with a genetic question on a particular animal, or hey, this animal you said is het pied, but it's not throwing pied babies, you lied to me, they can pull that original shed and say, no, I paid for the DNA registry on this animal, and this is the animal that I sold you. Why don't you send in a copy of your shed, and I'll pay to have that tested. And if they match, then something's screwed up. If they don't match, then you're lying, right? So it's one of those kind of cool things that we'll be able to do in the very near future. I mean, in the next couple months, I think we're talking about releasing it in October, where, if you're one of those breeders that wants that insurance policy on every baby that you sell, you can have it. I mean, we can register that animal and it'll be in our database forever. No matter how many transfers, no matter how many different owners it has, it can be tracked all the way back to its original breeder, and we'll be able to tell you this is the fingerprint genetically for that animal. There is no substitute. It's no different than a parentage test or in a court of law. I mean, we're talking about 99.99% accuracy at that point. So really, really cool stuff.
Yes, and not only would
the reason why I was distracted before that is before this, constricted reptiles. Dale Richards said he has lots of trick. Send me those sheds, please, Dale.
Yes.
Send us those every trick shed that you've got, please.
He's not far away. You save him up, Dale. I'll drive down and grab him from you.
Oh, is he in Virginia here too?
I think, if I remember right, I think he's in North Carolina.
My neighbor.
That's why I was distracted. I'm sorry I missed Morph Magic's question, but I saw that and I'm like, got to remember to circle back to Dale because we want those shits.
Yeah.
Yes. But yeah, with that, just to finish that up, not only would we be able to identify that individual that got registered for any kind of, like, if there was theft or anything like that, we would also be able to identify anything that's offspring from that individual with, like, times 10 to the minus blah, 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 like, 1 in 50 million match kind of a thing. Eventually, we'll be able to do all of that. So that will be another thing that will be really cool about that test.
That's cool. I'm getting like, I don't know. Y'all remember like Jerry Springer? I feel like that might be in poor taste to make a joke about that, but anyway, like, like you are it. They are the father. I don't know. Anyway, moving on. When you get this is from Johnny Cordero. When you get shed tests and they're labeled with other genes present, do you note the other genes on the test along with the gene you're looking for?
Do you understand that question, Shawn?
I do. Yeah.
You go ahead and answer it.
Yeah. So whenever you're ordering through Clutch or Morph Market, it asks you to please list the mutations that your snake may or may possibly have, along with what you're getting it tested for. If you leave that blank, we test your shed for the test that you bought, and then we throw it away. Because if we don't know what else is in it, we can't use it. However, if you list all of the other mutations that animal has or that you think that it might have, and it's a mutation that we think we can use for future research, we keep it. So we call it our Dewey Decimal System for snake sheds over at Ben's house. We have thousands of sheds now that we've held back that have mutations that we are interested in using to either refine current tests or develop new ones.
All right. We have from Samuel Ceresi. Do you think high OD and OD have any differences genetically based on what you have so far?
I don't have enough. I definitely had a good phone conversation with Ozzy about that, and he's interested in getting sheds to me. I actually found out, I knew that one of the animals I bought from Brian Sharp, and I asked him if that came from a high OD parent. He knew exactly which male. It was a super OD male kind of early on that he sold to Brian Sharp, and it did come from like his originator or one of either one of the originator high OD lines. So apparently I do have some high OD in my collection that I didn't know. So anyway, I've got a little bit, and Ozzy and I've been meaning to text or call him. That's another one. He's close enough. I think he's about a 5 or 6 hour drive. I'd be happy to go up and collect sheds, Ozzy, if you end up watching this. If not, I need to send you another message and let you know. That'd be great to come up and get some samples. And that would be a really good question. I think that's a really common one that I get. A lot of people would be interested in finding that out.
All right. Oh, actually, this is a really important question regarding the registry from Holdback Rack Podcast. Could you do the registry independently instead of through Clutch or Morph Market? Having them going through a third-party platform complicates things if the owner chooses not to use them anymore. And you have like just basic security, the more hands it's in, that sort of thing. Last part was added by me.
So I'm actually in discussions with both of our vendors for exactly this problem. There are a lot of kind of under the table business discussions that Ben and I don't have because they're just things that I think about that I think are going to need to happen. And so I mentioned it to our software partners, and this is one of the big ones. So both partners are working on a certificate to certify that animal that will be transferable in between one owner and another. However, the way they're currently designed, obviously, if one person purchases a test on the Clutch system, but then moves it over to Morph Market, there's no easy way to do that. So I am trying to get them to agree to allow us to be the database host for that data. So what that would mean is no, you would still have to purchase through Clutch or Morph Market. However, we would be the people who are actually holding that information. And when there's a transfer from one system to another, we would have to do it for you, if that makes sense, or from one vendor to another. Because they both agree that that's going to become a problem in the future, especially as DNA registry becomes a thing. As you point out, right now they're not easily transferable. And the only way to solve that is for us to hold it. So you're on the right track. We're actually thinking along those lines. That's about the only way that's going to be to solve it, is that we actually hold those records and we sort of send the information out to whichever vendor is currently holding that animal. And that will require some kind of partnership between the two as far as identifying numbers or whatever. So anyways,
we're working on it. Sounds like Shawn's thinking about it. That's the part that I don't think very much about.
Yeah, so that's what I do. That's the stuff that I work on. I've already been in talks with Sebastian over at Clutch and over at Morph Market with that exact problem, because I can see it coming.
Yeah. From Zeus's Serpents, have you been able to look at any snakes with a paradox to see if anything is genetic? We actually did an episode kind of about that. I don't know how many episodes ago.
Yeah. So I actually have the best paradox sample I've gotten. Somebody sent me a picture. They have a pretty good chunk of a shed, and it has very clear patches. So I'll be able to do 2 or 3 tests in each patch and see if I can see anything specific, whether it's a morph difference or a sex difference. I've had some talks with people about potentially doing that with their paradox animals, but this is the first time I have a shed on the way. So that'll be interesting. I don't know for sure. Theoretically, they should have a different genotype in those different patches, and the test should be able to pick it up. But since the same blood is flowing everywhere in that animal, what all makes it to the shed? I just don't know if I'm going to see like something in between, like if one patch is albino and the other patch is het albino. I don't know for sure if a test will be able to tell those apart, or if they'll both kind of look in between, not quite like a het and not quite like a albino. I don't know, but we're going to play with it.
Yeah. We'd love to hear an update on it, even if there isn't much like a way for us to see through your eyes as the geneticist. That would be a cool episode, I think.
Yeah.
Let us know if y'all are interested in that sort of thing for the future. We have Spoonman or Spoonman ball pythons. Are the results kept so that if we have tested now and the DGC or DGD genes are discovered, we'll be able to go back to our results and see if they're there?
So right now, we're just testing for DGA and DGB, so that you wouldn't have any results for anything other than DGA and DGB, because we have to actually tell the sequencing. We have to tell it where to sequence, and we don't know where C and D are. So you would have to send in another shed. Now, if it's, you just got your results for DGA and DGB, this next week on this next run, I get you your results. 2 weeks later I have a test for DGC, then I might be able to just run that for you because I still have it. But if it's been a month or two, then most likely that DNA is gone, and it would be difficult and time consuming for me to go back and find sheds. So more than likely you'll need to send in another shed. But yeah, that all will change when we start doing the DNA registry, like we were just talking about. That way you would just send in one shed and you don't have to worry about it from then on. Anytime we come out with a new test, you can just, push a button or send a message, let us know, and we can run that and tell you if you're positive or negative for whatever that new test is. You won't have to send in another shed. So that'll be a big plus for that test.
Yeah.
I'm reading a little bit ahead and a little further down. New York, NY, I'm assuming that's New York Powerballs. I think you should do the panel on a submission because a person might want to check for another gene in the previous submitted shed. Yes, that's exactly why the DNA registry software that we've been working on since the beginning was developed. If you guys pay for the registry, which will take the place of the panel in the future, we have an electronic fingerprint of your animal that we can go back and check for new test results anytime. Right now, if we're not in a position of your shed anymore, and usually we won't be within a couple of months, and again, unless it has other genes that we're interested in, we have to get a completely brand new shed and rerun the test anytime you want a new test on it, or if there's we find out something new like a DGC or DGD sort of a gene. So yeah, it's the exact same question, just two different people. That's the entire purpose, the number one purpose. Actually, there's lots of things you can use it for, but the biggest one is the registry will allow you to check. And by the way, within a day or two, you say, hey, I need results for these two new tests. Here's my payment, whatever we decide that's going to cost. And within a couple of days, you'll have your results instead of 2 or 3 weeks. So
yes.
So same answer, two different questions.
All right. Let's see. We have from Lee Hingle, any tests for puzzle coming soon?
Very soon. Yes, that's one as soon as we hear back. So Monarch, Puzzle, VPI, Xanthic, TSK, Xanthic, those 4. As soon as we get data back, I should be able to design and validate right away. And so unless I get bad luck on 1 or 2 of them, I would be very surprised if I don't get at least 2 of them and maybe all 4 of them in the next month.
Yeah. And how about the dark phase of TSK Xanthic? Asked by Tracy Sweat or Tracy Sweet.
I honestly don't know much about that. People, all I can see is what people call them when they how they notate what morph they have. And so if people start telling me there's a difference, I can run that as, something different. Or once we have the TSK Xanthic test working well, then you could send me ones that you believe are this darker phase and are not the darker phase. And I can just see if I see a difference with the test. But really, I just have to go on what people have sent in, and they've just said TSK Xanthic, pet or homozygous, and I haven't seen where people have talked about a darker phase. So that would be more complicated. And
yeah,
I have the samples. I can test it out. I can find out.
Yeah. And Tracy goes on to talk about how there are a few people out there who have these unusual TSK Xanthics that as it ages, it gets darker and darker. It makes me curious if it's somehow a polygenic thing or something else entirely, or just because that is, I guess if that doesn't happen to normal TSKs, that is interesting.
There's something in boas that's like that. They get a lot darker with age. That's really cool. I can't remember what it's called, but yeah, I would love to help figure that out. That'd be a great project.
Yeah. We have from, let's see, Samuel Ceresi again. We have anything in the works for Calico?
Yes. Yeah, I have some Calico stuff going. I did actually have an attempt at Calico, and it didn't look great. So if you have Calicos and could get, a few more to me, the more Calico sheds I've got, the better. I think I have a decent amount, but more is better.
All right. Oh, we just got an answer for the dark phase TSKs. When you mentioned something in BOA was like that. IMG, increasing melanin gene. I never realized that's what IMG meant. I just never thought, but that's perfect. It explains exactly what that is. Very nice.
I've seen some pretty cool ones that are super dark at reptile shows. They're very cool looking.
Yeah. Let's see, and then JD apparently has history of dark phase TSKs. I mean, once we have that test, I'd imagine then, if people do start sending in these differentiated sheds, we might be able to sharpen that point a bit, I guess.
Yeah.
Let's see, from Adam Olin, when do we get genetic testing for other reptile species? Which we've mentioned a bit as well.
Yeah. We are accepting sheds, getting sheds in for some species now. It really depends, first of all, on getting enough sheds in. Second of all, it depends on how much genetic information there is for that species or closely related species already in the academic literature. So for ball python, for example, we had to do our own whole genome sequencing because the Burmese python whole genome sequencing, first of all, wasn't that great and not close enough for what we wanted. Like Dr. Siddell mentioned in her papers, she had some difficulties getting the information she needed from the Burmese python genome. So there's some things that can slow us down. But the good thing that we have on our side after working through some ball python morphs is now we have an idea where to look in other species. So like when we start working with hognoses, we have an idea where to look for albinos or lavender albinos or some of the other morphs that are similar to ball python morphs. We can kind of cheat a little, and we might not have to do that whole genome sequencing that costs tens of thousands of dollars and takes quite a bit of time. So anyway, there's several things that go into it, and it's definitely months to years to get, like we mentioned a little bit earlier with ball pythons. It took a good year and a half or so for us to start churning out stuff for ball pythons. It's not a fast thing. It takes a lot of time and money.
Ideally, the best way to do it is the way we did it with ball pythons, which is to get a wild type male and female. And in our case, we were lucky enough to get wild caught individuals with like locality data, so we knew what we were getting. From that, as Dr. Ben was saying, we can kind of extrapolate backwards some of the more common genes that, maybe albinism or xanthics or things that you see across multiple species, that maybe they'll be in the same general area, but particularly things like corn snakes have so many different mutations, and some of them appear to be fairly complex. That's probably going to require a whole genome sequencing. And the last one we did was $35,000 for the ball python. I mean,
something like that.
Right.
Wow.
And that's just the wild type. So we would literally, honestly, we would make a trip out of it. Ben and I would go field collect some corn snakes probably until we get a pair that we're happy with, and then send those in and have the whole genome. I mean, the full-on genetic how to build a corn snake from the ground up, using its DNA. Sorry, I timed out. It's just super expensive, but that's the best way to go. Right now, we're not planning on doing that with any other species. We're hoping to be able to back into a few of the more common mutations first, and then once we get there, then we'll spend the money on other stuff. But as Ben was saying, that the good thing is we've learned an awful lot with the ball python stuff. It really should help us be pointed in the right direction on some of the other species. Pythons even more so, but even with the colubrids, so many of these things are very similar, if not on the exact same chromosome, just in a different animal. So
yeah. Adam, the same person, goes on to ask about leopard geckos. Now, well, lizard species, lizard and geckos are a lot more. Well, they're different from the snake species, but do you think that there are similar things going on genetically with some of these morphs?
Yeah, I mean, there's things in chickens and mice and stuff that, helped Dr. Siddell figure out where to look for yellow belly and albino and stuff. So yeah, I mean, what's happening in a snake is very likely the same thing as what's happening in a leopard gecko or in a monitor lizard or in a giraffe. I mean, a lot of the time it's the exact same gene that's mutated.
Yeah.
And so once we know that information, especially if we know it's the same in humans and mice and chickens, then it's most likely going to be the same in a snake or a lizard as well. So, but yeah, we would happily accept leopard gecko. We would love to work on leopard gecko genetic tests as well.
Yeah.
Hey, quick
Just
shout out.
know,
Sorry, Ben. Sorry.
I was just going to say, just know we can't do sex determination on leopard geckos. Anything that's temperature dependent sex determination, a genetic test won't work.
Immediately after Adam's question, Chad just said he likes seeing what we're doing. Chad's a good friend of ours and very helpful in what we do. And I just wanted to give Chad a shout out. If you guys aren't already sending him your samples for RNA, for NIDO testing and things like that, do so. He deserves the business, and the guy's been a big help to us, especially in the very beginning. So Chad, thanks, brother. We really appreciate you.
Hi,
Yes,
Chad.
Chad.
All right. And we have from NY Powerballs asking about the monsoon gene.
Yeah, so we mentioned that a little bit earlier. That's one that
right,
we
yeah.
are very close on. We got several sheds this last week that came through. And so that one, unfortunately, since we still need to do the sequencing, it's still going to be about 4 months out. But we do have at least a good amount of sheds now to work on that.
Yeah. We also have, misclicked. This was also a question on Facebook from earlier today from Adam, presumably Polish last name. I'm not going to butcher. When should we expect tests for cryptic? And as you know, which locus causes clown? Shouldn't it be quite easy to discover and develop? And any news about tests for morphs within blue-eyed leucistic, black-eyed leucistic complexes? So 3 different questions. Some we've already touched on, but take it away, Ben.
Yeah, so cryptic, I took a stab at cryptic because I thought I had enough that it might work, and it didn't. So I'm still working on that. Cryptic is coming. For those of you that don't know, if you have animals have to either be het clown or het cryptic, or if you have animals you're just trying to tell whether they're cryptic or krypton, then our clown test can help you be able to use process of elimination and know what you've got. But yeah, for a cryptic specific test or a het cryptic test, that's fairly close. I hope in the next couple of months we'll have that. Black-eyed Lucy and blue-eyed Lucy, we are the closest. We have the most sheds for Mojave and fire. So those will be the first out of those 2 complexes that we'll get. And we are pretty close on those. I would imagine the next month or 2. Those are ones I already have sequenced. It's just down to analysis and design and getting a test to work properly. So those ones I'm pretty close on.
Awesome. Thanks, Adam. So that's actually all we have in our rolling comments right now and from Facebook earlier. So do we have anything on Instagram? Any other questions from there?
Let's see. So Last Corn Snakes asks, how do you prioritize what tests you work on? It really comes down to what sheds we get. So if we have a whole bunch of sheds for a specific morph, it moves to the top of the list. The most important one for me personally were clown and pied, and pied had already been done by the group in Canada. And so clown was the first one. I really pushed hard. That's why we had a whole bunch in the first sequencing run we did. But after that, it's just been what we have sheds for. Whatever we have the most sheds for, we start working on that. There's a lot of people that asked for DG, and we got lots of sheds in. So that was one that I was at the top of my list for months and months. And then we finally found out why that was such a pain in the butt. But yeah, it's really sheds. So if there's a morph that you'd love to have a test for that we don't have one yet, and especially if it's one we haven't mentioned today, talk to people. If you have 2 or 3 and you can talk to 4 or 5 friends and they all have 2 or 3, you get it coordinated so those sheds come in. That can be the difference in getting it designed in the next month or 2 versus, another year down the road. Or if you can contact someone, has a whole bunch of them and say, hey, you're close to Ben, he'll come collect them, or I'm close to you, I'll come collect them, or, pretty please, could you collect these over the next couple of months? That's really what we need is sheds, and we'll make it happen.
Yeah, I feel like we all kind of know like that one kind of reclusive old guy who's been doing breeding these species for 20, 30 years. They're not online. They're just doing their own thing. Go find them, bring them a beer, and then say, hey, let me show you how to do this. Get them to send in some sheds for us. And from Spoonman Ball Pythons, when is the next DG result dump?
So next weekend, we'll have a whole bunch more DG data. So yeah, if you sent in for a DG test before, what day was that, Shawn? Like mid last week?
Up until mid last week, yeah. I'd have to go back and look at the spreadsheet. I'm not at the office now. I'm in my basement at home. So, yeah, mid last week, Tuesday, I think of last week is when we got everything packed up and on its way.
So as
So
long as it was received, if you have a received by date from Morph Market or Clutch, and it was received before kind of mid last week, then it's probably going to be in this group of results. And then hopefully about every 3 weeks, 2 to 3 weeks, we'll keep running more runs. And as far as the ones that people sent in for the free testing, we will just have to fit those in when we can. So there's no guarantee when those results will come, but we definitely will get them. It's just a question of when they'll fit on a run.
right now I'm trying to get 26 samples to fill a plate on DG on just the research samples that people are sending in, the free tests that we're offering. So I can send Ben a fully prepped, this is all DG matched with pictures and email addresses, so that he knows it's separate from our paid customers and it's a research run. And I've got enough to fill my first plate, so I'll probably do that this week and get it off to him with next week's paid shipments. And then I suspect we're going to be probably sending one every couple weeks over the next 2 months or so, because I get 4 or 5 envelopes a day full of the free DG testing offers that we did. And thankfully, everybody's been pretty cool about sending homozygous and 100% het animals. I think I shook a few apples out of the tree when I had to remind everybody in the email that I sent out with instructions. This is not to determine if your animal is DG. This is to determine what type of DG you have. So don't send me possets.
Yes.
And so far, everybody's been great. Like I said, I think I've got almost 100 sheds now, and they're all really good research possibility sheds, I guess. It's the best way to say it.
That's great. We have from Jacob Ledbetter, Sybil Serpents, or Jacob Ledbetter of Sybil Serpents. Any update on Monarch?
So Monarch is one of the 4 that we should have in the next month or 2.
Yeah. And then just a last response from Adam Olin. It's hard to do geckos when they eat. It's hard to get their sheds when they eat their sheds, which I actually hadn't even considered. And that's Godspeed gecko gecko breeders trying to get sheds.
And sometimes I know they'll kind of have some stuck shed like around their toes or tail base or something like that. And even if you send small pieces, really the first thing we do with sheds to get the DNA out is we break them down. So if they're in small pieces, that's totally fine. You just send 5 or 6 small pieces or 10 or 12 if you've got it. That works too. It doesn't have to be one solid piece the size of a quarter. It can be 20 small pieces. Spread
the word. We did a post about this 6 or 8 weeks ago where I posted a picture of a strip tube. I mean, a little pinhead size piece is all I take. You send me a giant ball python shed and I take a little pinhead sample. That's it. So if that's all you're pulling is a little sample like that off of the toe of a gecko, that's fine. As long as it's good, clean, dry shed, it'll still work.
Good to know.
Yeah. Just try to give us like 3 or 4 in case we need to come back and do another run. But even if it's 3 or 4 really small pieces, most likely that'll be enough.
Yep.
Perfect. And this one from Holdback Rack Podcast. Is there any way you can start accepting oral swabs for DNA sampling so we don't have to wait for sheds?
I would be happy to test that out if you wanted to try that out. Just send me a message. I guess probably through Facebook or Instagram or through the contact on the website. But just let me know if you or if someone else wants to send in like 3 or 4 swabs where you know the answer. So like say it's for sex determination and a ball python or whatever. They just need to be ones you know the answer, and then I can run it and then see if that works or not. I prefer myself what I do when I need a whole bunch of samples of something. Like when we were going to validate the black pastel and Enchi tests, I've got a bunch of black pastel and Enchi animals. And so I do scale clips. I flip them over and cut off the ventral scale. I just get a shaving off of the edge of a ventral scale. It's literally like the size of an eyelash. and I do it over a black poster board so that I can see it. If it shoots, a foot or something, it falls on that poster board. And then I put that into a Ziploc bag, and wherever it settles in there, I circle it so I can find it fast. I like doing that, and that works really well, but you need to have steady hands and maybe somebody helping you. And it's not the easiest thing. And obviously there's a chance if you don't have steady hands, you could hurt your snake. So it might not be good for everyone, and certainly not good for small snakes.
Yeah.
But yeah, if you want to try an oral swab, just make sure they haven't been housed with another snake, because that would be another potential for cross-contamination. What were you going to say, Shawn?
Just you and I have talked about this before, and we would want to test it on probably a few hundred different animals before we actually do this. But I've actually, from the business standpoint, talked to one of the two major reptile shipping companies about possibly being a fulfillment center for that sort of thing. So maybe in the future you guys order a shed test, but you don't want to wait for the animal to shed. So you pay a $5 fee or something, and it sends an email over to this company that puts together a kit for you and mails it to you so you can do a swab or something like that. So if we can get the swabs to work with the tests that we're currently running, and there's really no reason to think that they wouldn't work, but we would want to test it a lot before we do this. But we've already kind of thought about it, and the idea actually comes from Chad. He does that with a lot of his bird samples, where people order online and they're just automatically shipped a swab kit, and they send back whatever they need to send back in a nice clean little plastic bag with everything all buttoned up and nice and neat. And that would sure be nice from our standpoint, especially for those folks that don't want to wait 2 months or whatever for their animal to shed. They can spend an extra $5 and have a kit shipped to them in a couple of days. They can turn around and have a swab back to us within a week. Yeah, I'd like to do that. We're already talking to one of the big reptile shipping companies to fulfill those for us.
Yeah. Oh, here's an interesting one from Jamie Rising of Phoenix. Jamie of Rising Phoenix Reptiles. What about DNA testing from embryos? Like if you just had some hatched, like the embryonic fluid after they hatch.
Yeah, I mean that should work great. Blood works really well. I've done many blood spots. Any kind of piece of paper or paper towel, doesn't really matter. Just be clean. Don't let it have been sitting in your reptile room for a month. But if it's a clean piece of paper and you just that baby just is about to hatch out, and you just get a little bit of blood from a egg vein and get it on the piece of paper, let that dry, send that in, that works. But you just got to make sure for sure which baby goes with that egg, and a lot of people don't keep track of that.
Yeah.
Yeah. What were you going to say, Shawn?
I know that the bird companies that do sex determination on pet parrots and all kinds of other birds actually can pull it right from the eggshell. You send a piece of the eggshell from that animal in, and they can pull the DNA out of that. I don't see why that wouldn't work for us too. But like Ben said, you better be really good about knowing exactly which egg. I mean, at that point, you're probably talking about tearing the eggs completely apart from each other and making sure there's quite a bit of distance in between them, so that when the animal hatch, they don't cross-contaminate by sliming all over each other. So it'd be a little more complicated, but is it doable? Yeah, but there's a potential for serious cross-contamination going on there.
Yeah. Lori Torini brings up an interesting point about collecting sheds collecting sheds being probably the least intrusive manner to get DNA samples, meaning there should be no stress to the snakes. These other methods are likely or possible to cause some level of distress to the animal, which, especially if you're dealing with like hatchlings and stuff. So your mileage may vary. Be careful, I would say.
Yeah, if I'm not in a hurry, a shed is by far the easiest, because whether I throw it away or put a piece in the bag, the way I personally do it when I have animals I want to collect sheds from is I pre-label the Ziploc bag with the animal ID, and I just put it right next to its cage. And so I know, as I'm looking in cages and stuff, I know which ones I want to get sheds from. And as soon as I get that shed, it goes in the Ziploc bag, and I just leave it unzipped and put it somewhere for it to dry. And then as soon as it's dried, I shut it. And that way it's away from that cage. I know I don't need to collect a shed from that animal anymore. So if I have a clutch hatch out that are posset clown, I just put a pre-labeled bag by those 7 or 8 animals. And as soon as they shed goes in the bag, gets dried, shut it, and then it's ready to test.
Yeah. We shared a post from, I think, JDH Reptiles earlier where they sent us a picture of their racks. They just have like bags and like a sheet of paper like on the platform right beside their racks. Like if they've shed, there's a piece of shed there laid out, drying. It's perfect. And it's just like that all the way down every single rack. It's perfect. And yeah, it is the least stress on the animals.
The guys at Canova did a good one too. It wasn't Justin or Summer, but it was the guys that actually work in the snake room. They go through their whole process about how they label each animal, what tests they're going to want, and then they take the shed and literally just hang it on the outside of the drawer of that animal so that it has an opportunity to dry. And then they put the shed and the sticky and they separate it into the different tests that they need for what particular. That they did a really cool video too. That was, I think, 6 or 8 weeks ago. That was a really good one too. Their system is pretty spot on.
Yeah. Let's see here. I unfortunately cannot read the characters there, but from this person, they're asking, I've been wanting to ask you this for a long time. Do you think black pastel and cinnamon are the same gene that's been line bred differently?
So I have run the black pastel test on many black pastels by this point. So this, once again, I'm just going off of what keepers have told me their animal is. So I've run, dozens and dozens, might be close to 200 known black pastel animals or super black pastel. And it's always given me a positive for black pastels. I have run like 6 or 7 cinnamons that are personally mine. Half of them were mine, and then the other half were from other people that labeled them as a cinnamon, and they come up negative. So it is at least a little different. It could be like asphalt and gravel, where it's a mutation in the same gene, but it's a slightly different place. And if it's in a slightly different place, my test will be negative. I haven't chased that down for cinnamon yet to be able to tell you if it's, close or the same gene, or, I haven't done that yet. But that's something we certainly will do.
Yeah. All right.
But it's not identical. I can tell you that.
Good to know. Glad we got to answer that for you. Do we have anything on Instagram? Because unless we get more comments here, either on Facebook or YouTube, that's all I've got.
Yeah. So Hades Herbs, he says, my pied het DG boy is doing great. Did you have yours tested? I think, is this Tim? Are you Hades Herbs?
Mm-hmm.
I don't know for sure the answer to that. I thought it might be one that I know what he's talking about, but there's one that I purchased that was a DG het pied. Derek, I don't know for sure which one you're talking about, Derek. You have to give me a little more info.
Mm-hmm.
But yeah, I did purchase a DG het clown male, and he did test homozygous for A and B. So Tim Slotkey, that was from Tim.
Ooh.
I don't remember the name of his. I think it's Slotkey's Serpents.
Yeah, Slotkey's Select Serpents, I believe.
Yeah. Oh, he's talking to someone else on IG. That's why.
Oh, okay. Yeah, no, I get caught by that too. They'll have conversations with each other, and I'm just like, wait, what is that? Oh, no, they're conversing. They're socializing. Perfect.
69 Skydiver 69 asks, any news or updates on boas? I've had a few sheds coming in, not enough. I think that Hair Hollow Farm, I think she's holdback rack. I think she was going to send in some boa sheds. I think she might still be on here. She can say if she's got that on the way yet, but I'm hoping that we'll at least be able to do the sharp and call strain tests so people can be able to have one or the other. But yeah, we'll see.
Yeah,
I don't have enough sheds yet to do any boa tests. I need more sheds.
Yeah, let your boa friends know. We actually did an episode that might have been number 3 where we had Travis join us. So we had 2 geneticists here, or they're here with us, talking about a whole different things related to the albino gene, and boas especially were a topic of that. So it was a few weeks back, but go check it out. You might get some answers in there if you haven't seen it already. We have from Amber Thomas. Have you guys figured out if red axanthic is similar to black pastel?
I really don't get very many samples that are red axanthic or het red axanthic. That's not a lot coming through. I actually have a clutch hatching out right now that has got het red axanthic in it. And I do have a few animals. So I'll have to run some of those with the black pastel test. I would assume it's going to be like cinnamon, where it will be negative for the black pastel, because it's different enough. It's not an identical mutation, but it would not surprise me at all if it ends up being like the yellow belly complex, where it's different mutations that are in the same gene.
Yeah. Let's see. This is from actually, do you guys, so this is from Holdback Rack. I'm not sure which gene they're referring to. They have a ton. They were trying to wait until they have more of those. Do you want those sheds to just be sent in now, or just to get the party started?
Honestly, it's kind of up to you. I don't have enough to do anything right now. So if you are kind of to the end, this is all you're going to get, send them. If you think you're going to still collect some more, then yeah, definitely just collect some more. And especially if you can talk to somebody else and say, hey, I've got a bunch of sharp and call stuff that I'm going to be sending in. You should be keeping yours, and let me know when you're going to send in. We'll send them all in at the same time. Once I have over 20, then I'll probably send some for sequencing. So if you're to a point where you already have over 20, I don't know if you have that many, but then it would be worth sending them in. But otherwise, that's totally fine to collect a few more and get that done.
Yeah, and emphasis on like finding other breeders who maybe have different lines, because the more genetic diversity we have in these, the better it is for Ben when he's sequencing and trying to find where he can identify these two different lines, or two different traits. Also from Jacob Ledbetter of Sybil Serpents. If I send in a shed that is unconfirmed to be an actual visual DG or just a low quality one, or pos het, would you be able to identify that it's het or visual? Or does it just show the homozygous or heterozygous for DG A or B?
So it's going to be the latter. So our test can tell you what the DNA says, but since it's a polygenic trait, and part of whether it's visual or not depends on at least one other gene that we don't have a test for. I can't tell you for sure if your animal is visual or not. I guess that I could tell you, what percent of the ones that have that specific genotype, what percent of those breeders have reported as being visuals. But it's definitely a test on the DNA, on what the DNA says. It's not a test on the trait itself, how it looks on the outside, the phenotype. It's a genotype test. And so I can tell you whether it's het or homozygous for DG A and B. And I think that kind of what you're getting at, if it's a question in your mind whether this is a low quality visual or not, I think that the research we're doing right now with the sheds from you all, where we have sheds and then we have pictures, so we'll have a genotype and a phenotype to look at, I think that we'll be able to help people be able to make that call better here pretty soon. And then also, as we get more information on DG, potential other genes like a DGC, then it may get to a point where we'll be able to predict that better, whether it will be visual or not. But we're not there yet. It's still in process.
All right. We have, Dale is asking, well, he says he has a couple of leopards and het leopards on the boas, and is asking if y'all are coming down to Daytona, which, yes, Shawn's going to be there.
Yeah, bring them.
Yeah.
Any sheds you got for new tests, bring them to Daytona, bring them to Pomona, bring them to the October Tinley. That's great. Or
any RBC Arlington, I'll usually be at one of those two. The fall show I'm usually not at because it always falls like smack in the middle of bow season for elk, and I'm usually in the mountains. But the spring Arlington show, I'm definitely going to be there. Arlington's not even an hour from where I live, so
awesome. Let's see. Scales and Tails Breeding is asking, or it's just mentioning, it just occurred to me that the stickers I made and sent with my last test were clown, and that was the first test you made. That's neat. Hope y'all like them. And Shawn, I know you have a wall that you keep like the stickers and stuff that
it's actually it's a little bit of a misnomer. It's one side of my white refrigerator where I keep like reagents and stuff and freezer. So yeah, it's all
got it timed out there.
Colored up now with all the people's stickers that they've been sending me. So keep sending them. I'm having a good time slapping them up there. And once I get a whole bunch, I'll take a photo that we can show everybody.
Yes, please. I want to post that picture. Let's see. We have from Endless Morphs. Are you working on testing for acid confusion and static being the gene and determining if those are the same? Let's see. And Blitz, Trick, Hurricane.
Yeah, those are ones I'm very interested in. Confusion is the one I have the most sheds for. I personally have a few acids, but not very many, like 3 or 4 or something like that. So yeah, if you all have acid, static, or confusion, especially acid and static, I don't have very many sheds from those at all. Send them in. But yeah, confusion, I think we have a chance at figuring that out in the next couple of months. We're fairly close. And then Blitz, Trick, and Hurricane. Hurricane, I have the most sheds from, but it's still not enough for me to design from. So I need more Blitz, Trick, and Hurricane sheds. So definitely send them in if you've got them.
Yeah, tell your friends. And Jamie again from Rising Phoenix Reptiles. Will you be at the Schaumburg or just Tinley for the Midwest?
For me personally, probably just Tinley. I don't know that I'll be able to make it that far again. I can't remember which month that's in, but yeah, Shawn and I will have to talk about that. Both of us have talked about how we need to lay that out, but we haven't done a good job of that yet.
I don't know how far Schaumburg is from my house.
Close to Chicago, I think.
It's a 14-hour drive for me to get to Tinley from where I live. And flying up there and renting a car and stuff is doable, but we're all the shows we want to do, if we both try and do every single one of them, we're not leaving a whole lot of time to do other stuff like research and run tests. Remember, it's just the 3 of us and like one part-timer who helps check sheds in. So shows are really kind of high up on our priority list, but right now we're not a big enough company to be able to do it all. We'd rather make sure we hit the main ones that are easier for us to hit, or like we said earlier, our anniversary show, and keep our customer service levels up and our test times down. And that means we got to stay home and work.
Yeah.
Yeah. So people are saying Schaumburg. Yeah, Schaumburg's right by Tinley, and it's the end of July. So yeah, I definitely this time of the year is going to be very busy. We're going to have the most sheds coming in July, August, and September. So
maybe
any shows I have to travel for that are in July, August, September. October is a little bit pushing it, but usually babies have mostly slowed down by October. But yeah, a July show, I don't think I'll ever make that.
Yeah, to give everybody an idea, we're not even in our first year of business yet, so we just don't know what to expect. But we sort of figured that as we approach baby season, we would get busier. And this month, we're on track to have the best month we've ever had by far, probably 2 or 3x over since the day we opened. And that's not to brag. I think it's just a function of the fact that there's a lot of babies hatching and a lot of people. I'm seeing a lot of orders coming through that are paid that I'm going to expect to see sheds coming in over the next 2 or 3 months. And I think that's going to continue all the way through probably September and into early October before we start to trend back down again. So these months, it's really hard for us to go anywhere because it's super, super busy season. I'm super busy receiving and checking in sheds and prepping sheds. And I know Ben is, and he's running the actual tests on several of them and doing the research. And yeah, like he mentioned at the very beginning of the show, our last run was the biggest one we've ever crammed into one run, right? Like 700 samples in one run. That's a lot.
Yeah.
Now, that wasn't all paid samples. A lot of it was research, but still, that's the most we've ever done. And that's just an inkling of what's coming. So we have to be careful about travel plans during the busiest time of the year.
Oh, yeah. What about St. Louis in November? Asked by Dustin Tinsley.
That's definitely better than July for sure. I probably won't make it this year. I don't know. That's probably still a pretty good drive for you too, right, Shawn?
Yeah, if it's slower though, I don't mind flying and renting a car. I can't not be here when I can see as much happening. And it's so hard for us because we're such a new business. We don't know what we don't know. So it's hard for me to plan ahead when, I hate to buy tickets and rent a hotel room and get a car all prepped and ready only to not go because we're too busy and I've got to stay here. So we're learning a lot this first year. This first busy season, I've said many times in these calls, is going to kind of be an indicator for how we plan the next few years. If we are doing 5 and 6x the volume what we were doing in March and April, then the fall and the summer and fall is just not a great time for us to travel a lot. It's just not.
And if things keep going like they have been so far this month for the next 3 months, then we might be able to hire another person or two. And that makes it so Shawn and I can focus on other things. I don't need to be the one running the tests. We can certainly hire someone that already has some experience and they can do the part that I don't have to be the one there. And then Shawn and I can make it to more shows and do more fun, kind of connecting with you all. And then I can also design faster too. If I'm not running samples, I can be designing new tests quicker.
This next 3 months will definitely tell the tale because, excuse me, goodness, I like going to shows. I want to go to more. And I know Ben feels the same. And this is the first time that actually we have a business that can pay for it, so that we don't have to pay for it out of our own pockets, right? Yeah. So that's pretty cool. But yeah, this first year it's just tough. We're being careful and cautious and trying not to spend money where we're not sure it's going to be a waste. But yeah, if we put up the numbers we think we're going to be able to put up just in this first year, and we have 3 times as many tests this time next year, there'll be more people on board and a little more freedom to go around for travel.
Yeah. Question from Hades Herps. Do y'all have a list or a site where we can get info on what sheds y'all need? Or ones that we're maybe looking for specifically? We
have an anti-list. We have the list of ones we don't need. Those are the tests we have designed. Anything else, it's on the hit list. Right now, we don't really need Monarch, TSK, VPI, Xanthic, and then what's the other one? Puzzle. So those ones are already, we have pretty good numbers and they're already sequenced. But yeah, most anything else, if we don't have a test for it, it's worth collecting sheds. And if you think you can get, a couple dozen or something between you or a couple of people and, send us a message through Facebook or Instagram. Those are probably the messages that we check the most, and email. But yeah, just send us a message, say, hey, I really do a lot of paint stuff. There's 2 or 3 of us will collect paint sheds for you over the next couple of months. I just want to make sure that's something that you can, hit the gas on. If you tell me that over the next couple of months, I can expect to get, 20 or 30 paint sheds. As soon as they, get to me, I'll be hitting the gas on that. So that's for any more. If you think you can get several, give me a heads up and we'll make it happen as fast as we can.
Yeah. Ultimately, and correct me if I'm wrong, guys, but from what I understand, the tests that we develop are the things that basically get sent to us. It's you guys, the tests that you're interested in that get sent into Rare Genetics. That's what we're going to test for. So, we can put out a call for whatever, but if it's not popular, then we're not going to get sheds for it. Granted, it can be helpful to focus on one shed, one test that we need more of, Desert Ghost, but that's different. It's Desert Ghost. So
Elite Exotics BP on Instagram is talking about Monsoon again. So we know from tonight, Monsoon is a hot one. Many people have asked about Monsoon. So right now is a great time. You say you have 3 visuals. Please do send those in to me if you can, because we talked about this a little bit earlier, but I'm going to be sending in sheds for Monsoon here pretty quick. So if I can get them in the next couple of weeks, especially, then they'll for sure go on that run. So that would be great.
Yeah. And Dale from Constricted Reptiles is asking if you're still setting up at shows like Raleigh or Charlotte in North Carolina.
I would if I could.
I know we tried this past year, and I think we were able to make one, maybe. I think, but
yeah. It's gotten so much busier with the DNA testing that I haven't made it to the more local shows.
Mm-hmm.
But yeah, that's a good point. Limey does have a lot of paint. I'll have to I'll have to message him and see if he's willing to either let me come down or if he'll collect some. So y'all and most of North Carolina is close enough that I wouldn't mind driving down there and going through, and I don't mind going through cages and picking out sheds. And if you'll let me do scale clips, I'll do scale clips. We can get a whole bunch in 4 or 5 hours.
There you go. All right. What other questions do we have from Instagram?
I don't see. Still more monsoon talk. We've been going pretty long too.
Yeah. Actually, we're
all the questions.
Hour and 40 minutes now.
I was gonna say it's gonna be dark here soon, and I still have to go feed all the farm animals. So.
Oh no. Okay. Yeah. Maybe.
Gotta let Shawn go take care of the animals.
Yeah.
All the ducks and the chickens and all that are probably literally at the front door waiting.
Oh, I'm sure. Dad, where's the food?
Yep. And I've got a wife and 4 kids, so the 4 kids that need attention, the wife might get mad at me if I'm too much later. So
well, that's fair. Well, all right. I guess do the usual, like, subscribe, comment below. Definitely check out our social media. We've got Facebook and Instagram. Of course, our YouTube if you're watching there. Check out our website. We had a little blip last week where it was down. It is back up. Check out our merch. And am I missing any of the normal? Oh, Shawn.
Just real quickly, we went so long. There's a lot of people watching now that probably weren't watching earlier.
Oh, yeah.
2 things. One, send us a shout out to Caddy Strike. She's got a professional MMA bout that we're sponsoring this weekend. We wish her luck. Send her a PM or something. Let her know we sent you and that we're wishing her luck. And then don't forget, it's not really a contest so much as we're asking you guys for creative ideas for artwork for new shirts that we can give away and sell and things like that at shows. And there'll be stuff we have on our merch store, but then there's going to be some special funny stuff like the DNA test results. Reptile nerd, check the box. Anything creative, any kind of creative any creative logos, thank you, artwork or anything like that, even if it's just not really well drawn, but it's just a good idea, I'll pay to get it professionally drawn. But please send us those ideas, that creative fun stuff. I love that stuff, and I love seeing it on shirts. I think the best shirt in the reptile industry ever made was the King Cobra eating the albino ball python. So anything creative that you think goes well with a business like ours, please send it to us. I'll make sure you get a free t-shirt, some free testing, or even more if it's a really good design that I don't have to do anything with and I can just use it as is. You'll obviously get credit. That's it from me. I'm out. That was all an hour and a half ago that we talked about that. So I just want to make sure it's still here. Please do that.
And Dale, you're in Florida. I forgot you drove that far to come up to the Charlotte shows.
Yeah.
If I can make it down to Florida, I'll have to reach out to you.
Yeah. All right. Well, let's call that a wrap, you guys. And thank you everybody for showing up. Outro time. And let's go ahead and do it in 3, 2, 1. Later, guys.
Holy crap, that was long.